Welcome back, Bio 121. We're still in Chapter 5. We're looking at plasma membranes, and we've gone to this location where plasma membranes are selectively permeable. And we need to now start thinking about the fact that you've learned that all plasma membranes are made from amphipathic molecules that will interact with things such as ions on the outside or the inside of the cell. And if they're going to interact with them, how can we actually move materials between the two sides of the membrane? Let me draw that for you. Okay, let's just draw that as, here we go. Here's the outside of a cell. Here's the nucleus. I'm going to draw them smaller so you can get the general idea, right? And let's imagine, if you will, we have some ion inside of a cell, really small concentration. We have a really large concentration of that ion outside here. And that means the cell is going to be like, I don't want you inside of here. And that means it's going to have to expend energy to push it against its concentration gradient out of the cell. And that would be what we call active transport. Passive transport, of course, could be something as simple as requiring no energy at all. In the earlier example of me saying carbon dioxide would just simply diffuse by passive transport out through the phospholipid bilayer because it's small enough to do that. Okay? Now, having told you that, let's see what happens when we try to develop this a little bit more. Passive transport here is diffusion is going to occur from that area of high concentration to an area of low concentration. You'll see I haven't lied to you. Small non-polar materials such as oxygen, carbon dioxide, other gases like lipid hormones such as nitric oxide pass easily through biological membranes. Okay? This is where you want to put together, say, a little set of flashcards for yourself where you can look at what's going to affect diffusion rates, because you can imagine that what I'm going to do is I'm going to create a scenario where if you either increase or decrease pressure, something's going to happen. If you increase or decrease the solubility of something, it's going to either diffuse faster or diffuse less quickly. Okay, temperature is going to cause molecules to move faster, but that doesn't necessarily mean they'll diffuse faster. Okay, they may move more quickly, but that doesn't mean they're going to. Okay. Generally speaking, it does, but for yucks and giggles, you need to keep an open mind. Okay, the first type of major types of transport is facilitated transport, where you build a specific channel or a carrier protein, and this allows that material to move through the cell. The one example I gave you a moment ago would be aquaporins that allowed water to move in and out of a cell. The ones your textbook focuses on are, of course, ions. Or in this case, I'm just going to draw sodium. Okay, sodium. And remember, if this is the inside of the cell, you may not even understand this yet, but we should probably make sure you understand it, right, before we keep going. Why do I keep drawing a small representation of sodium here and a big representation of sodium here? One of the requirements of being a living organism is to maintain homeostasis. Your textbook barely touches that in the beginning of Chapter 1, but all of the work that this cell is doing right now is to use facilitated transport to move ions. And in this case, it's supposed to be passive transport, so this isn't really a good example. But imagine, if you will, we could use something like, glucose, C6H12O6, right? Where the channel, when activated properly through the protein insulin, will now move into the cell. No energy required because the concentration of glucose outside of the cell is always higher than the concentration of glucose inside of the cell because you're constantly breaking it down. Okay. So, now, how do channel proteins actually work? When they work, they're going to allow some specific molecule to move through. This is a decent representation. It's a little confusing by the other things here. This should not be allowed anywhere near that, right? But the idea, of course, is that some are open all the time. Some are only open when they receive an appropriate signal, sort of like that insulin example I gave you a moment ago. But the idea, of course, is you actually have to allow materials in, and they will then transfer out. Carrier proteins do something a little bit different. Okay, the glucose transporter I was just telling you about, glucose binds shape will change and will now be pushed in. What they're leaving out of this cartoon is there are other things that are happening at the same time. So you get the idea that it's a carrier protein, but the carrier really just does one thing, and that is adding specificity. Now, osmosis is this idea that materials, in this case water, will diffuse across the membrane such that it will move to an area of lower concentration of water, right? So what we mean is, if you could actually have a semi-permeable membrane here, with time, let's imagine this is what happened. There's more water on this side of the membrane, less water on that side of the membrane, and water would move from this space to that space there, and this would be the consequence. Okay, you'd actually have more water, and now what's most important that most textbooks leave out is, okay, what's the big deal? Well, the big deal is this. Water is going to change its location. Can your cell accommodate that change in location? Fact of the matter is it cannot. So what Mother Nature does is she exerts an osmotic force or an osmotic pressure, and that osmotic pressure literally pushes this back down such that they come close to equilibrium, okay, in such a way that the shape of the cell can be maintained. Okay? Because if you have too much expansion here, too much water moving into this space, you'll rip the cell apart by what's called hypotonic lysis. And if you have too little water inside here, the cell will crenate. You're looking for that happy medium between these two spaces. But osmosis, along with diffusion, are the two primary forces that drive movement of molecules inside of your body. Now, the consequence, of course, of all of this is what we call tonicity or tonicity, right? When solubles with different osmolarities are separated by a membrane to water, but not the solute, water moves. This is the consequence I was just telling you about. A hypertonic solution versus a hypotonic solution and an isotonic solution. And dollars to donuts, they're going to use red blood cells to explain this to you. Look at that. If we move red blood cells into a hypertonic solution, what's going to happen is, notice the cells have crenated where all the water has now moved out to dilute this solution. So it's closer to what's inside the cells. Isotonic is roughly the same amount of water moves in and water moves out. Hypotonic, just as dangerous as hypertonic. All the water inside this space here moves into those red blood cells there, and notice they're no longer their correct size, they can't do their job. Notice, hypertonic cells no longer their correct size, they can't do their job. The reason why patients are given an isotonic saline solution when they're admitted into the hospital, regardless of what they're at in terms of health status, is to maintain the likelihood that red blood cells can deliver oxygen with regard to, or regardless to, anything else that's happening inside the body. Inside of a plant, remember inside the plant cell chapter, the plant cell wall construction discussion we had earlier, the cell literally would change shape here. It's still attached to that cell wall, but notice when the plant's dehydrated, like the ones right outside my window again, okay, the water is all gone out of that vacuole. It's moved into this space to try and maintain that turgor pressure, whereas when some people kill their plants with too much water, what they've done, of course, is squish the cell from the inside. Okay? Both are bad for your plants. Active transport is where you need to spend some time thinking, right? Active transport is this idea that anytime an ion or molecule, such as glucose, are transported into a membrane, it's moving through a concentration gradient. And there are two types of transport. There's either primary active transport or secondary active transport. Glucose on the whole doesn't have to use primary active or secondary active transport for the facilitated responses I told you before. In fact, some cells inside your body don't have any regulation for glucose at all. Your liver and your central nervous system do not require any form of regulation for glucose to get in and out of their cells. It's just taught so that you understand that glucose will move down its concentration gradient. Wham! Now, there's a consequence to this, and that's sort of the cartoon I was drawing for you earlier. Let's draw a cell again. I want now for you guys to understand what the consequence is of actually having a small sodium here and a large sodium there. And let's imagine you've got a big potassium here and you have a small potassium there. Well, the consequence is going to be this. As a result of some other molecules inside of the cell, namely phosphates, what ends up happening, and this is where life actually begins in terms of our ability to respond to things, you can begin to take advantage of this. And what you end up doing is you create a resting membrane potential at the surface of the cell. And this goes all the way around, so I'm just going to draw a blue line all the way around, representing that resting membrane potential of a positive charge on the outside. And let's go yellow on the inside, just because it'll look visually, you can't even see that. What are you talking about? Let's go, I don't know, orange? There we go, orange, is that better? It is, look at that, it's more like a brown than it is an orange, right? But the idea, of course, is now you have a negatively charged environment on the inside of the membrane. And this becomes an on and off switch relative to the amount of ions in charge. And remember, Mother Nature is going to work fervently to maintain this concentration gradient there, and that extra effort to maintain that concentration gradient there, as well as many others. But the take-home message is this. Electrochemical gradients are going to arise as a result of this partitioning. The consequence of actually doing that, though, okay, is you now have an electrical gradient at the membrane of the cell. And you might recall the beginning of chapter 5, I drew for you those 4 different tissue types. And remember, I segregated them by excitability and non-excitability. Remember, muscle cells and nervous system tissue is excitable. And the generation of this charge at this membrane is what will give it its excitability. Okay, that's actually how it gets that functionality, is the point that I'm trying to make. Okay, so we're going to move those ions. Fantastic. And when we move those ions, you're going to see that as we're moving them, you have a consequence. You can actually have some things moving in one direction. Those would be uniporters. Symporters are glucose and, say, sodium, or glucose and calcium can move into a cell at the same time. Or let's say we're going to move sodium in one direction and hydrogen protons in another direction. Uniporter, one direction on one molecule. Symporter, 2 different molecules using the same transporter, both are required. Likewise, for the antiporter to function, you require both A and B in the appropriate concentration to move outward. And that's kind of really hard to regulate if you're thinking about it, right? So when we're trying to wrap the rest of this up, let's think about how you actually demonstrate the electromotive force at the membrane itself. And this is what we're talking about here is, imagine, if you will, you had 3 sodium ions you had to pump out, and you have to pump them out. This is a sodium-potassium ATPase, and this is what actually is expressed on the surface of all of your cells. And to move this against its concentration gradient, notice what happened here. 3 sodium ions bound inside the transporter. It was phosphorylated by ATP, and that ATP now pushed the energy change here, pushed those ions out, and it changed the shape of the channel. In doing so, it could bind 2 potassium ions. And when it does this, it removes that phosphate and the ADP, and now the potassium ions can actually move in. So this is, generally speaking, the easiest way to remember is sodium-potassium ATPases are primary active transport, and they require ATP to push 3 sodium ions out. The energy returned back into the system changes the shape of the channel, pushing that phosphate off, and that phosphate now allows, because it's been removed, allows for the the potassium ions to move down their concentration gradient. This is a really important idea, so I'm hoping you spend some time thinking about how I might ask questions like this, about this on an exam. Now the secondary active transport piece here is, they're showing glucose moving in, right? And what they're doing, of course, is they're saying there's a concentration gradient, and they're saying some other molecules moving with it. Let's say for yucks and giggles, it's going to be sodium or calcium, and this is going to come in, and both of these will come in at the same time. But notice what happens. It's a primary movement of glucose. It's secondary for the sodium or the calcium to move in as well. Bulk transport is how we get large amounts of things in and out of a cell. So you order a pizza and you eat more than 2 pieces. That's bulk transport. You eat more and more than you actually need. But that's an oversimplification. How we get things in and out of a cell are called endocytosis and exocytosis. And there are 3 types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. These all work together. Open up your mouth big, stuff in 2 pieces of pizza. Pinocytosis. Sip some water with amino acids inside of it and other materials. Receptor-mediated endocytosis would be if you literally required a molecule to signal to the cell, it's okay to bring that material in now. Sort of think of it as a manifest check before the contents of a cargo ship are allowed to enter the port. Same idea. There's a YouTube video for you to go watch. In fact, you're going to see, I'm not quite sure how I'm going to show it to you inside the lab. We're probably going to do some sort of osmosis lab for you to think about how it might work. But here's an example of some phagocytic event occurring. And notice what's happening. We're literally using the membrane to create, inside some organisms, we're going to call it a vacuole. It's a vesicle is what you're making. A large particle comes in, you encapsulate it with plasma membrane, and now the cell knows this is inside the cell. Now, you can do this with pinocytosis because small amounts of ions, that sort of sipping of that glass of water I was just talking about, can now move in. And maybe these materials come in and the fluid stays there and the ions move out. Depends on what the cell actually needs. And here's receptor-mediated endocytosis, where you have a specific set of proteins at the membrane. And once the specific ligand binds to the receptor, notice what happens. Clathrin literally causes a vesicle to form. That coated vesicle now allows for that material to move into the cell. There's a whole bunch of stuff that happens after this, but for right now, this is a pretty good place to stop and start and think about what this means. Exocytosis is a little bit different, right? And you need to think about it in the sense that whatever the cell is making or needs to get rid of, it will now move to the membrane, fuse to the membrane, and I want you to think about what this might actually look like. Let's pick a different color here. Let's go red. And I want you to think about this piece of the membrane right there. What it actually does is it hones in on that spot right there. But when it does that, what it actually does is it now causes everything inside of here to now be exposed to the outside. And that's what they're trying to represent for you here. This here was once that there. And this is actually how it works. Mother Nature makes materials, puts them inside vesicles. The vesicles bind to the plasma membrane. The plasma membrane then exocytoses that material out of the cell. So those are the major thoughts in the first exam, chapters 1 through 5, right? Where what we want to do is we want to think primarily about how it is we now have made a functioning cell without telling you about any one cell. Now that's how you want to study this material. You want to think to yourself, what are the mechanisms and definitions of mechanisms that I've learned, and how could they help me describe how life can now start to resemble this dynamic system?